Hello friends, I am Dr. Firuza Parekh. I am the Director of the Fertility Jaslok International Fertility Center at Jaslok Hospital and Research Center in Mumbai. First of all, I would like to thank my co-authors, Dr. Dhananjay Kulkarni and Dr. Arundhati Athalye, who helped me in preparing this commentary. Today, I would like to discuss my commentary on Casanova's paper, which is currently in press. The title of the paper is Double-Stranded Sperm DNA Damage Measured with Neutral Comet is a Cause of Delay in Embryo Development and Can Impair Implantation Rates. Before we go into the heart of the paper, we realize that DNA is the repository of genetic information, and if damage to it occurs and it is not repaired, this can lead to mutations. The sperm DNA is tightly compacted by unique associations between the DNA, the nuclear matrix and the sperm nuclear proteins. The sperm nucleus is compacted and stabilized by the DNA strands taking a toroidal or donut shape. This protects the sperm DNA from external stress and subsequent DNA breakage. Indeed, there is a threshold of DNA damage beyond which embryo development and pregnancy are impaired. Casanova's group made a very important observation regarding the impact of double-stranded sperm DNA fragmentation on embryo quality and morphokinetics as compared to single-stranded sperm DNA fragmentation. There are several methods used to analyze sperm DNA fragmentation. These include the tunnel method with microscopy, sperm chromatin structure assay, also called SCSA, then sperm chromatin dispersion called SCD method, and the comet assay that the authors have used. The alkaline comet assay detects mainly single-stranded sperm DNA fragmentation, while the nuclear comet assay can detect double-stranded sperm DNA damage. Sperm DNA damage can occur because of intrinsic factors such as protamine deficiency, DNA packaging defects, advanced paternal age, and abortive apoptosis. External factors contributing to sperm DNA fragmentation including heat, radiation, gonadotoxic therapy, smoking, and the presence of varicocele. These factors can cause oxidative damage leading to DNA fragmentation. This particular paper was designed as an observational double-blind prospective study in which 196 embryos from 43 infertile couples were implanted. The essence of their paper was that double-stranded sperm DNA fragmentation significantly lowers embryo implantation rates as compared to low double-stranded DNA fragmentation or high single-stranded sperm DNA fragmentation. They observed the morphokinetics of embryos and concluded that double-stranded DNA fragmentation caused a delay in embryo development and impaired implantation, while single-stranded DNA fragmentation did not significantly affect embryo growth and implantation. Double-stranded sperm DNA fragmentation caused a delay in all stages of embryo development, including extrusion of the second polar body and morula formation, coinciding with embryo DNA checkpoint activation. In contrast, single-stranded sperm DNA fragmentation caused its major effect at the pronuclear stage. However, in this case, the embryo's kinetics were restored at all the subsequent stages, and this did not significantly impair growth and implantation. In an earlier paper, the same group have reported an association of double-stranded sperm DNA fragmentation and unexplained recurrent miscarriage. Studies by Simon et al. In 2013 have shown that sperm DNA damage has a negative correlation to the time to pregnancy. Such damage also increases embryo cleavage time and miscarriage rates. A review study by Zini and Libman in 2006 has also reported an increase in birth defects in children where the father had increased sperm DNA fragmentation. Hence, the integrity of the paternal genome is of paramount importance in the initiation and maintenance of a viable pregnancy. Sperm DNA fragmentation is a major contributor to male infertility. On another note, our team has examined the effect of diabetes on sperm DNA fragmentation. We presented our findings at the 2013 All India Obstetrics and Gynecology Conference, where we showed that sperm apoptosis in diabetic men was 18% as compared to controls where it was 6%. We are currently working on examining the effect of advancing paternal age on sperm apoptosis. Our preliminary data suggests that lower sperm DNA fragmentation was observed in the 25 to 30 years age group compared to the highest DNA fragmentation which was observed in the 35 to 40 years age group. This singular test has the potential to effectively predict reproductive potential and should be used to counsel couples with male factor infertility. In fact, this test is believed to have the potential to be used as a very important diagnostic tool as it has the lowest variability of all semen parameters. I thank you for your patient hearing.